Good day, ma'am. We are the group 1. So these are my group mates, Julumay Galicanta, Hannah Shane Bustillo, Guardito Bajo III, Nurhana Naho, and yours truly, J. Ann Tao of Narid. We will be presenting to you our study about the seafood industry entitled Major Foodborne Pathogens in Fish and Fish Products. The study aims in keeping fish and fish products safe as more people in Europe are eating them. The growth of industry and farming can make the water where the fish live dirty, affecting both fish health and our safety. We will talk about a certain bacteria that cause the problem and how they impact the water where the fish live. Fish in natural environment can carry bacteria from contaminated water, affecting their gills, skin, and digestive tract. While healthy fish are typically sterile, certain organs may have bacteria. The pathogenic bacteria like Campylobacter, Salmonella, Yersinia, and the Listeria monocytes are present in fish, causing their health risks. But not all bacteria can cause the outbreaks. But the Listeria monocytogenes, Vibrio species, Salmonella, Yersinia, and the Clostridium botulinum are asserting due to crucial despite and nutritional benefits of fish consumption. Join us to understand how these relate to make sure the fish that we eat are safe and won't make us sick.
The research review aims to know the prevalence of pathogens in the contaminated area around the globe. The research review focuses on 5 bacteria that are causes diseases, not just in fish but also in humans when they eat contaminated fish or fish product. To start with review species is common in various aquatic environments, and these bacteria are often observed in skins, gills, and intestinal tracts of fish or shellfish. It is defined as gram-negative astrogenous rods that are straight or have single review curve. This bacteria can contaminate fish and fish products using improper handling, long-time transport, evisceration, and also cross-contamination from raw materials. Vibrio species can cause 3 types of infections: gastrointestinal, wound, and blood. So symptoms of it is gastrointestinal vibrios include watery diarrhea, abdominal cramps, nausea, vomiting, and or fever. Vibrio bacteria can be destroyed by cooking fish to an internal temperature of 145 degrees Fahrenheit for 15 seconds. It will cook fish, especially in summer months. Also, Salmonella cerevisiae prevalence in its organisms na mesophilic, or it means that bacteria defined as organisms that grow at moderate temperature with an optimum growth temperature in its suitable range from 35 to 37 degrees as low as 2 degrees Celsius. And like as 54 degrees Celsius, it never relies on oxygens, unlike aerobic. And mostly survive in low moisture. Food in fish is influenced by factors like water contamination, inadequate feed, and poor hygiene during capture handling, and transportation affecting both live and caught fish.
So next, we have the Clostridium botulinum. They are anaerobic, meaning they live and grow in low oxygen conditions. Bacteria form protective spores when conditions for survival are poor. The spore has a hard protective coating that encases the key of the bacterium and layers of protective membranes. Botulinum toxins are ingested through improperly processed food in which the bacteria or the spores survive. Then, grow and produce toxins. Though many of foodborne intoxication, human botulisms can also be caused by intestinal infections, usually affect the weakness of the muscles that controls the eyes, face, mouth, and throat, often spread in neck, arms, torso, and legs. Botulisms also can weaken the muscles involved in breathing, which can lead to difficulty breathing and even death. Next, we have the Listeria monocytogenes. It's a species of pathogenic or disease-causing bacteria that can be found in moist environments, soil, water, decaying vegetations, and animals, and can survive and even grow under refrigeration and other food preservation measures. It can grow at low temperatures such as in the refrigerator, 1, and can grow up to 120 to 141 days. Imagine how long these bacteria survive. When people eat food contaminated with Listeria monocytogenes, they may develop a disease called listeriosis. Moreover, at risk of listeriosis include pregnant women, newborns, older adults, and people with weakened immune systems. On the same time, 2 to 21 days, growth conditions of this bacteria temperature range is from 1 to 40 degrees Celsius or 34 to 113 degrees Fahrenheit. The refrigerator, 1, and can grow up to 120 to 141 days. Imagine how long these bacteria survive. When people eat food contaminated with Listeria monocytogenes, they may develop a disease called listeriosis. Moreover, at risk of listeriosis include pregnant women, newborns, older adults, and people with weakened immune systems. On the same time, 2 to 21 days, growth conditions of this bacteria temperature range is from 1 to 40 degrees Celsius or 34 to 113 degrees Fahrenheit. Optimum temperature from 30 to 37 degrees Celsius or 86 to 98.6 degrees Fahrenheit. Can be resistant to the effects of freezing, drying, and heat. So lastly, we have the Yersinia species. It is a group of gram-negative, non-spore-forming, oxidase-negative, catalase-positive, lactose-negative, and facultative anaerobic organisms that grow conditions temperature range up to 0 to 45 degrees Celsius or 32 to 113 degrees Fahrenheit. The optimum temperatures of this species or the bacteria is from 37 degrees Celsius or in Fahrenheit it is 98.6 degrees. The potential hydrogen range is tolerant of alkaline environments, or simply means that not acidic surroundings because if it's acidic, they can survive due to its tolerance level. So here are some common symptoms in children like fever, abdominal pain, and diarrhea, which is often bloody. Symptoms typically develop 4 to 7 days after exposures and may last 1 to 3 weeks or longer. In older children and adults, right side abdominal pain and fever may be the predominant symptoms and may be confused with appendicitis. All these pathogens mentioned are harmful not just for the health and the fishes, but also for the consumers who consume it. Poor hygiene and proper handling are considered as the top factors for fast bacterial reproduction. Hence, proper practices like making sure to clean the apparatus to be used, proper storage, and most especially is the maintenance for good fish farm conditions.
So, in this research, there are tables that presents the data on bacterial contamination for various seafood in different regions and years. It includes the numbers of samples, contamination percentage, bacterial strains found, and contamination locations. So, for example, the first entry in the table reveals the findings from a study in India conducted between 2003 to 2006 on shrimp. Out of 86 samples, there were 23 contaminated, or 26% of the 86 samples. 26.7% of the 86 samples. And bacterial strains like Bereli, Brenderu, and Tipi were identified. So, the contamination occurred at both landing centers and retail outlets. The following entries follow a similar format, presenting information on contamination in different seafoods types like clams, oysters, cuttlefish, octopus, and squid across various regions from India, Iran, and Egypt. And the years they were identified were from 1990 to 1992, 2003 to 2006, and 2009 to 2011. So the study is important because it helps us understand the contamination of seafood, which is harmful for food safety. But by knowing where and when these pathogens is more common in seafood, we can take steps to protect public health. This information guides the development of safety measures to ensure that the seafood we consume is safe and free from harmful bacteria.
So they study major foodborne pathogen in fish and in fish product. So was made by Alexander Novoslokdis, Margarita Terentseva, Inga Eisenberga, Olga Basina, Vadim Barkevics, and Ivar Bernjins, and was published in May 31, 2015. So the purpose of the study is to understand and highlight the potential contamination of fish with harmful bacteria such as Salmonella, Listeria, monocytogenes, Vibrio, species plural. So Clostridium, Botulinum, and Yersinia species plural, the study emphasized that contamination can occur through two factors like human activities, dirty white, and poor hygiene during the capture and handling of fish. This contamination is not only a threat to the health of the fish population but also raises concern for public health since people can get sick from eating contaminated fish and fish products. So the study suggests that following good hygiene practices is crucial to prevent contamination and ensure the safety of fish